How has prenatal diagnosis evolved from ultrasounds to genomic sequencing? Imagine being able to see inside your womb with more detail than ever before. Over the past 70 years, prenatal diagnosis has changed a lot, giving families more information about their baby's health before birth. It all started with simple images and has grown into complex genetic analysis. This progress helps parents decide on screening options and prepare for their child's needs. In the early days, ultrasound technology was introduced in the 1970s. It used sound waves to create pictures of the developing fetus. These images help doctors spot major physical issues like missing parts or abnormal growth. Early ultrasounds were basic, just measuring the baby's head or body, but they quickly improved to show clearer images. Today, ultrasounds are routine for checking fetal development, confirming pregnancy dates, and identifying structural problems. Then came the discovery of chromosomes under a microscope in the 1960s. This led to karyotyping, a test where doctors take a small sample of amniotic fluid or placental tissue to look at the baby's chromosomes. It could detect conditions like Down syndrome, which is caused by an extra copy of chromosome 21. But this method was invasive and carried a small chance of miscarriage. It only found large chromosome changes, not smaller genetic issues. The late 1990s brought a new breakthrough. Scientists found fetal DNA floating in a mother's blood. This discovery led to non-invasive prenatal testing, or NIPT, which started in the late 2000s. NIPT uses a simple blood sample from the mother to analyze cell-free fetal DNA. It can screen 4 common chromosomal conditions like Down syndrome, Edwards syndrome, and Potthouse syndrome with high accuracy. Because it is non-invasive, it has become a popular choice for many families, especially those at higher risk. More recently, the field has moved into the era of genomic sequencing. Instead of just counting chromosomes, tests like whole exome sequencing and whole genome sequencing look at thousands of genes at once. These advanced tests became available in the early 2010s. They are especially useful when ultrasounds show unexplained abnormalities. These tests can identify rare genetic disorders caused by single gene mutations, such as certain skeletal or neurodevelopmental conditions. They provide a much more detailed picture of the baby's genetic makeup. For families, this evolution means more options and better information. Ultrasounds help monitor growth and anatomy, while karyotyping and NIPT screen for chromosomal issues. Genomic sequencing is used when other tests don't give clear answers, especially if there are unusual findings on ultrasound. These advances help parents make informed decisions about screening and prepare for their baby's health needs. It is important to understand that each of these methods has its role. Ultrasounds are safe, quick, and essential for visual checks. Karyotyping is good for large chromosomal changes. NIPT offers a safe, early screening option. Genomic sequencing provides detailed genetic information when needed. As technology continues to improve, prenatal diagnosis will become even more precise, helping families plan and support their children from the very start.